In 1922, archaeologists cracked open a sealed chamber inside the tomb of Tutankhamun. Gold, jewels, sacred artifacts preserved for over 3,000 years. And tucked among the treasures meant to accompany a pharaoh into the afterlife, a small jar of tiny black seeds. They weren't decorative, they were medicine. The same seeds had been described in ancient Egyptian medical texts as a remedy for headaches, infections, toothaches, and digestive illness. Across the Arabian Peninsula, a tradition passed down for 14 centuries recorded a statement attributed to the Prophet Muhammad. Use this black seed, it has a cure for every disease except death. That single sentence made Nigella sativa one of the most recognized natural remedies in the Islamic world. But here's what most people don't know. Modern science didn't dismiss it. Modern science confirmed it. Over 1,300 peer-reviewed studies have been published on thymoquinone, the active compound inside the seed. Anti-inflammatory, antimicrobial, blood sugar regulation, liver protection, immune modulation. The data is public, the studies are accessible, the seed costs less than $10 a pound, and yet most people in the Western world have never heard its name. The question is, why? The story does not begin in Egypt, it begins earlier. Cultivation of Nigella sativa dates back over 3,000 years to the Assyrians of ancient Mesopotamia. They called it tin tir. They swallowed it to settle the stomach and applied it to the skin for rashes, bites, and inflammation. From the Tigris and Euphrates, the seed traveled south into Egypt, west into Greece, and east into India. By the time it reached the pharaohs, it was considered essential enough to bury alongside gold. A Hittite flask containing Nigella sativa seeds was recovered from a site in Turkey dating to the 2nd millennium BCE. The seed was not regional, it was civilizational. And by the 21st century, over 1,300 peer-reviewed studies would document what three empires already knew. But the pharmaceutical industry was spending $1.4 trillion a year selling synthetic alternatives to what the Assyrians had figured out with a seed and a mortar. In the 1st century of the Common Era, the Greek physician Dioscorides documented Nigella sativa under the name melanthon. He prescribed it for headaches, nasal congestion, toothaches, and intestinal parasites. Hippocrates, the man Western medicine calls its founder, recorded the same plant in his therapeutic texts. 500 years later, the physician Avicenna, whose canon of medicine remained the primary medical reference for European universities for over 600 years, described Nigella sativa as a treatment for difficulty breathing. He prescribed it for fatigue, lethargy, and general debility. In India, the seed entered the Unani system of medicine, which teaches that health depends on the balance of 4 humors: blood, phlegm, black bile, and yellow bile. Nigella sativa was prescribed to restore equilibrium. In Arabic, the seed is called Habat al-Baraka, the seed of blessing. 3 continents, 3 medical traditions, 3 independent conclusions that the same seed was essential to human health. And then modernity arrived and replaced all 3 traditions with patentable molecules. Here is what the laboratories found when they finally opened it. Most pharmaceutical drugs contain a single active molecule. This seed contains over 100. Nigella sativa seeds hold over 100 identified bioactive compounds. The oil fraction comprises 32 to 40% of the seed's total weight. That oil is rich in linoleic acid, oleic acid, and palmitic acid, essential fatty acids that the human body cannot produce on its own. But the compound that has generated the most scientific attention is thymoquinone. It is the primary active constituent of Nigella sativa volatile oil. Chemically, it is 2-isopropyl 5-methyl-1,4-benzoquinone, a name designed to make your eyes glaze over, which is convenient for the people who would rather you did not pay attention. Hold one of these seeds between your fingers. It is smaller than a grain of rice. Black, angular, with a faint peppery smell that sharpens when you crush it. Inside that shell is a compound that has demonstrated, in laboratory and animal models, the ability to modulate immune response, reduce inflammation, protect liver cells, lower blood sugar, inhibit microbial growth, and selectively trigger apoptosis in cancer cells while leaving healthy cells intact. One seed, 100 compounds, and a price tag that makes pharmaceutical executives look the other way. In the early 1990s, researchers at King Faisal University in Saudi Arabia began systematically testing what traditional healers had been prescribing for centuries. They isolated thymoquinone, measured its effects on immune cells, cancer cells, and inflamed tissue, and published what they found. Other laboratories followed, then more. What started as a handful of papers became a cascade. The research is not a handful of pilot studies from obscure journals. It is a body of evidence spanning decades, institutions, and countries. Over 1,300 peer-reviewed publications have investigated the pharmacological properties of thymoquinone and Nigella sativa extracts. The effects documented include antioxidant activity, where thymoquinone scavenges free radicals and protects cells from oxidative damage. Anti-inflammatory properties, where it inhibits the biosynthesis of prostaglandins, leukotrienes, and cyclooxygenase, the same pathways targeted by ibuprofen and aspirin. Immunomodulation, where it regulates the activity of T cells, B cells, macrophages, natural killer cells, and dendritic cells. Anti-cancer activity has been demonstrated in vitro against breast cancer, colon cancer, prostate cancer, pancreatic cancer, ovarian cancer, and head and neck squamous cell carcinoma. Thymoquinone induces apoptosis in cancer cells through multiple pathways, including generation of reactive oxygen species, DNA damage, and interference with the NF-kappa B signaling cascade. In animal models, it has shown selective cytotoxicity. It kills tumor cells while sparing healthy tissue. A 2025 comprehensive review published in a peer-reviewed pharmacology journal confirmed that thymoquinone exhibits antioxidant, anti-inflammatory, immune-stimulating, antiviral, and antibacterial effects in experimental settings. The review also noted that clinical research evaluating safety and efficacy in humans remains limited, partly due to thymoquinone's poor bioavailability and biological instability. That last detail matters. It is the lock on the gate. In 2024, a systematic review and meta-analysis covering 30 randomized controlled trials examined the effect of Nigella sativa supplementation on glycemic markers in adults. The results were published in Clinical Nutrition, ESPN. The pooled data showed a significant reduction in fasting blood sugar and hemoglobin A1C, the gold standard measurement of long-term blood sugar control. A separate meta-analysis of 16 randomized controlled trials, published in 2025, focused specifically on type 2 diabetic patients. In hospitals across Saudi Arabia, Iran, and Pakistan, patients received measured capsules of black seed oil alongside their standard medications. When the lab results came back, the numbers had moved. Black seed supplementation reduced fasting blood glucose by 21.43 milligrams per deciliter. It reduced HbA1c by 0.44 percentage points. It lowered total cholesterol by 18.80 milligrams per deciliter and LDL cholesterol by 19.53 milligrams per deciliter. The oil form, at doses above 1 gram per day, was more effective than the powdered seed. These are not marginal effects. A 0.44-point reduction in HbA1c is clinically meaningful. Pharmaceutical companies have built billion-dollar drug franchises on smaller improvements. Metformin, the most widely prescribed diabetes medication on Earth, typically reduces HbA1c by 1.0 to 1.5 percentage points. A seed that achieves one-third of that effect at a cost of less than $10 a pound should be headline news. It is not. Here is the bottleneck that keeps thymoquinone out of clinical medicine. The compound is hydrophobic. It dissolves poorly in water. It degrades rapidly in the body. It has low oral bioavailability, meaning much of what you swallow is broken down before it reaches your bloodstream. This is not an unsolvable problem. Pharmaceutical science has developed nanotechnology-based drug delivery systems specifically designed to overcome bioavailability limitations. Researchers have already encapsulated thymoquinone in PLGA nanoparticles with 97.5% efficiency, dramatically enhancing its anti-proliferative and anti-inflammatory effects. Under an electron microscope, these particles appear as perfect translucent spheres, each one a delivery vehicle engineered to carry thymoquinone past the digestive barriers that would otherwise destroy it. Liposomal formulations have maintained thymoquinone's anti-cancer activity while improving stability. Mesoporous silica spheres have achieved slow-release delivery that outperforms free thymoquinone in inducing cancer cell apoptosis. The technology exists, the delivery systems are proven, the compound works. But thymoquinone is not patentable. It is a naturally occurring molecule found in a seed that costs $10 a pound and grows in hardiness zones 3 through 10 across most of the world's temperate and tropical regions. You cannot patent nature. Developing a drug requires phase 1, 2, and 3 clinical trials costing hundreds of millions of dollars. No pharmaceutical company will invest that money in a compound they cannot exclusively own. As of 2025, only 2 phase 1 clinical trials have been conducted on thymoquinone in humans. 2. After 1,300 studies proving it works in the laboratory, the science is not the problem, the business model is. The United States Food and Drug Administration classifies Nigella sativa as generally recognized as safe, GRAS, for use as a spice, natural seasoning, or flavoring. You can buy it at any grocery store that carries Middle Eastern or South Asian ingredients. You can order it online for less than the price of a fast food meal. It is legal. It is available. It is recognized as safe by the same regulatory body that approves pharmaceutical drugs. But the FDA has not approved it as a treatment for any disease. And without that approval, no manufacturer can legally claim it treats, cures, or prevents any condition. This creates a regulatory paradox. The data exists, the mechanism is documented, the safety profile is established, but the seed cannot be marketed as medicine because no one has paid for the clinical trials that would allow the FDA to reclassify it. The pharmaceutical industry does not suppress Nigella sativa through conspiracy. It suppresses it through economics. The incentive structure of drug development requires patent protection to justify investment. A seed that anyone can grow, anyone can harvest, and anyone can sell for $10 a pound offers no return on a billion-dollar clinical trial pipeline. That is not conspiracy, that is the structure. And the structure is the suppression. Walk through the Khan el-Khalili market in Cairo, and you will find a vendor named Hassan, whose grandfather sold black seed from the same wooden stall during the British occupation. His great-grandmother mixed the seeds with honey for every ailment the family could not afford a doctor for. Hassan will tell you to crush the seeds fresh, fold them into raw honey, and eat them on an empty stomach before sunrise. He will not cite a meta-analysis. He will cite his grandmother. In India, Nigella sativa is called kalonji. It is pressed into naan bread. It seasons pickles and curries. It is a kitchen staple used daily by hundreds of millions of people. In Bangladesh, it is scattered over rice. In Turkey, it is baked into simit, the sesame-crusted bread ring sold on every street corner in Istanbul. Across the entire Muslim world, the prophetic tradition has ensured that black seed never disappeared from daily life. The statement attributed to Muhammad did not make the seed a curiosity, it made it a staple. 14 centuries of continuous daily use across a population of nearly 2 billion people represents the largest uncontrolled nutritional study in human history. The knowledge survived not in laboratories, but in kitchens. Not in clinical trials, but in family traditions. And it survived because it was never separated from the culture that used it. Nigella sativa seeds are available at Middle Eastern grocery stores, South Asian markets, health food shops, and online retailers across every Western country. A pound costs between $8 and $15, depending on source and quality. Cold-pressed black seed oil, the form with the highest concentration of thymoquinone, typically sells for $15 to $30 per bottle. You can add the seeds to bread, salads, yogurt, smoothies, or rice. You can crush them in a mortar and mix them with honey, exactly as Egyptian physicians prescribed 3,000 years ago. You can take a teaspoon of the oil daily, the dose most commonly studied in the meta-analyses, showing blood sugar and cholesterol improvements. One caution matters. Quality varies enormously. Look for cold-pressed, organic, unrefined black seed oil. The thymoquinone content in commercial oils ranges from less than 1% to over 5%. Higher concentrations correlate with stronger clinical effects. Standard culinary-grade seeds are food, not concentrated medicine, effective over time as a dietary staple, not as a single-dose treatment. No prescription required, no patent, no corporation in the supply chain. The seed is the product. The pattern is the same one you have seen with every plant in this series. A natural compound with documented therapeutic potential is sidelined because it cannot generate proprietary revenue. The structure of modern pharmaceutical development requires exclusivity. Without exclusivity, there is no investment. Without investment, there are no large-scale clinical trials. Without clinical trials, there is no FDA approval. Without FDA approval, no Western doctor will prescribe it. And without a prescription, the public is taught that the absence of approval means the absence of evidence. But the evidence is there. Over 1,300 studies, 30 randomized controlled trials on blood sugar alone. A compound that modulates immune cells, inhibits inflammatory pathways, and selectively kills cancer cells in the laboratory. A seed that 3 ancient civilizations independently classified as essential medicine. A food that 2 billion people still eat every day. The seed costs $10. The pharmaceutical alternative costs hundreds. The seed cannot be patented. The alternative can. That is not a medical decision. That is a financial one. The story of Nigella sativa is not about a seed. It is about what happens when healing cannot be monetized. For 3,000 years, this seed has traveled with every empire that touched it. The Assyrians carried it. The Egyptians buried it with their kings. The Greeks prescribed it. The Persians codified it. The prophet declared it essential. Indian healers balanced humors with it. And modern scientists, using the most advanced analytical tools ever built, confirmed what all of them already knew. The seed works. It has always worked. And the only reason most people in the Western world have never heard its name is that no one can charge you $500 a month for something you can grow in a window box. In 1922, a jar of black seeds was found in the tomb of a pharaoh who died 3,300 years ago. He believed they would protect him in the afterlife. In the centuries that followed, 3 civilizations built their medical traditions around the same seed. In the 21st century, 1,300 studies confirmed they were right. And still, most people in the West have never heard the name Nigella sativa. But now you have. If this vault opens something for you, subscribe to Nature Unearthed and hit the bell. Every like and every share preserves what they tried to erase. The next vault opens soon.